microbe, they sit on the cell wall, the antibiotics, and they disrupt the cell wall, making all the protoplasmic contents spill out of the cell, hence killing the cell or destroying the cell. Methanogenic bacteria are extracted from the cow dung or goat dung or the fecal matter of the ruminant animals, and from there they decompose the fecal matter further to produce CH4 or methane gas, which is also known as biogas, since it acts as a fuel. They live in colonies in root nodules of leguminous plants such as pea. All those cyanobacteria that are specialized in fixing atmospheric nitrogen are known as heterocyst cyanobacteria.
Thank you for watching this video.
Hello students, welcome to btosacademy. So let us go forward with our lesson of the 5 kingdom classification. In this class, we will discuss kingdom Monera, kingdom Protista and kingdom Fungi in details. Let us start with kingdom Monera. Now, kingdom Monera are all prokaryotes. Meaning they do not have a well-defined nucleus. Their genetic material is sprangled all over the cytoplasm known as the nucleoid. Also, they do not have well-defined membrane-bound organelles as we see in plant cells or animal cells, or even in cells of kingdom Protista or cells of kingdom Fungi. So, kingdom Monera can be divided into various classes. Now, what are those classes? The first class that comes under kingdom Monera are the mycoplasma, which we call it as PPLO or pleuropneumonia-like organisms. Now, these mycoplasma are the smallest of all living cells and are also the smallest living organisms on earth. They are also known as jokers of plant kingdom because during the 2 kingdom classification scheme, as we have studied earlier, Carolus Linnaeus placed mycoplasma into kingdom Plantae or along with plants. However, this is not true. Mycoplasma is a prokaryote, whereas plants are all eukaryotes. Since they do not have a well-defined cell wall, they are insensitive to antibiotics. Mycoplasma, if it enters our upper respiratory tract, it can cause various infections which have symptoms similar to pneumonia, like severe cough, cold, sore throat, breathing difficulties, and other symptoms associated with pneumonia. So they are known as pleuropneumonia-like organisms as they primarily attack the pleura or the covering of our lungs. Since they do not have a cell wall, since antibiotics, what they do, if this is a microbe, they sit on the cell wall, the antibiotics, and they disrupt the cell wall, making all the protoplasmic contents spill out of the cell, hence killing the cell or destroying the cell. But since they do not have a cell wall, the antibiotics do not have an attachment site, so insensitive to antibiotics. Antibiotics cannot kill mycoplasma. Also, they are anaerobes. They do not require oxygen for their sustenance and survival. Another class is Rickettsia and another class is Actinomycetes. Now, these are also very small microorganisms, unicellular prokaryotes. We do not need to study Rickettsia and Actinomycetes in details. What we need to study is the class archaebacteria and the class eubacteria. These archaebacteria and eubacteria encompass all the organisms which we commonly call as bacteria. Now, archaebacteria can be classified into 3 subclasses. What are those? First is the methanogens, then comes the halophiles, and then comes the thermoacidophiles. Coming to the methanogens, they are found in the digestive tract of ruminant animals such as goats, such as cows, such as buffaloes, etc. those animals that eat grass and plant material only, that is the primary consumers. We know that cows, goats, they all ruminate, right? They bring out the semi-digested food into their mouth from their stomach. They have food chamber stomach, rumen, reticulum, omasum, and ovomasum in the cows, and they bring out the food from that rumen part of their stomach, and they again chew it and again they re-ingest it. This is known as chewing of the cud. They do this to break down the food into smaller bits so that the cellulose in the food, the cellulose present in the plant material can be easily broken down. The methanogens actually does a fair share in breaking down of the cellulose of plant material, and in the process they also produce a gas called methane, which is also known as a biogas acting as a fuel. Methanogenic bacteria are extracted from the cow dung or goat dung or the fecal matter of the ruminant animals, and from there they decompose the fecal matter further to produce CH4 or methane gas, which is also known as biogas since it acts as a fuel. Coming to the halophiles, halophiles are present in extremely salty conditions where the salt content of the water is very high. Halophiles are exclusively uh, aquatic kind of archaebacteria. They are found in volcanic lakes, in mountainous regions, the lakes of the mountains, such as we have some in Himalayas and all, where the salt content is very high or the water is saline, and also in ocean water. These halophiles are very important factors in our oceanic food chain, and also they some halophiles are also photosynthetic. They can also produce a lot of oxygen that is essential in that aquatic environment. Coming to the thermoacidophiles, the thermoacidophiles are present in those conditions where it is very hot and the pH is very low, preferably in volcanic shafts and vents and in volcanic lakes. Thermoacidophiles are very important in genetic research. One example of thermoacidophile is the bacteria Thermus aquaticus. This Thermus aquaticus bacteria can tolerate temperatures of up to 100 degrees Celsius. They can tolerate boiling water, and they can tolerate pH up to 3.5 or even pH as low as pH 2. So, what is the use of this Thermus aquaticus? In this Thermus aquaticus bacteria, we extract an enzyme from this bacteria known as the TAC polymerase or TAQ polymerase, the Thermus aquaticus polymerase. Now, what does this TAC polymerase enzyme do? This TAC polymerase enzyme helps in the process of southern blotting. Now, what is southern blotting? Southern blotting is a forensic technique from where you can collect DNA from crime scenes, right? And you can multiply that DNA in order to zero in on the criminal. Now, for the PCR reaction, what we do, the polymerase chain reaction, where we multiply strands of DNA, if we have a 10 kilobase strand of DNA, we can amplify it using the PCR machine into a million base pair DNA, right? So this polymerase chain reaction is catalyzed by TAC polymerase, which we can extract from the thermoacidophilic Thermus aquaticus archaebacteria. So PCR is followed by comparing DNA strains of the various suspects in order to zero in with the criminal who has committed the crime. So this is southern blotting. So this bacteria is very important for forensic investigations. Archaebacteria are the oldest type of bacteria that we have. Since they are known as archae or primitive, and bacteria you all know it's bacteria only. So they are the primitive or the most primitive of all bacteria. Another class of bacteria is the eubacteria or the newly evolved bacteria. Newly evolved means they are not evolved 5 years or 10 years back. They also have evolved millions of years back, but in comparison with the archaebacteria, they are relatively new and a bit more evolved. So, eubacteria is further classified into two subclasses: the true bacteria and the cyanobacteria. We will first discuss the cyanobacteria. Cyanobacteria are also known as blue-green algae since they live in colonies similar to algae, and they are photosynthetic, they are autotroph. They produce their own food by photosynthesis. They have chloroplast, and some cyanobacteria like nostoc, like anabina, they can fix nitrogenous oxygen, the N2 gas present in our atmosphere, as salts of nitrates and nitrites in the soil, which helps plants to grow, which acts as a natural fertilizer. So they can also fix the atmospheric nitrogen into nitrates and nitrates in specialized cells called heterocyst cells. Now, cells by mean they are unicellular. So where are the cells coming? You must question this. See, they live in colonies in root nodules of leguminous plants such as pea. All those cyanobacteria that are specialized in fixing atmospheric nitrogen are known as heterocyst cyanobacteria. So, heterocyst cells. For example, nostoc and anabina. Coming to the true bacteria, well, they are autotrophic, they are heterotrophic, they are saprophytic, which we will discuss later. We can classify true bacteria primarily on the basis of their cell shape, the shape of their cells. First is coccus. Now, coccus means the bacteria live in colonies like this, like a bunch of grapes, and individual bacteria looks like a sphere, a ball, or a grape, and their colony is called coccus or cocci in plural. They live like a bunch of grapes. Their colony looks like this. Bacillus is the rod-shaped bacteria. The rod-shaped bacteria. Vibrio is the comma-shaped bacteria. Vibrio is the comma-shaped bacteria, and Spirilla, they are the spiral or spring-shaped bacteria. So, based on the shape, we can classify them into coccus, bacillus, vibrio, and spirilla. Also, the true bacteria have their cell wall composed of peptidoglycan and picoic acid. They have a cellulosic cell wall. Cellulose is not present in their cell wall. In addition to the cell wall, they have an external covering that encompasses or encapsulates the cell wall, known as glycocalyx or capsule, which covers the cell wall. It is a secondary mode of protection. Now, based on staining, bacteria can be classified into another two groups. This grouping was done by the Swedish scientist Hans Christian Gram, and the process was known as the Gram staining of bacteria. The stain he used is the crystal violet stain. It is a dark bluish violetish type of stain. He saw some bacteria, when stained, take up the color of the stain, they become blue colored or violet colored, whereas other bacteria, they do not take up any stain. The bacteria that take up the stain are known as gram-positive bacteria. For example, lactobacillus. For example, lactobacillus. Those bacteria that take up the stain have a single layered covering and they have picoic acid in their cell wall. Bacteria that do not take up the gram stain are known as gram negative bacteria. They have a triple layered covering and they do not have picoic acid in their cell wall. There are a lot of gram negative bacteria, and generally the bacteria that are very dangerous to us, that are pathogenic, that produces disease, such as Vibrio cholerae, such as like spirulina, bacteria like this, they are all gram negative bacteria. So mostly the pathogenic bacteria are all gram negative. Lactobacillus is also very important bacteria. It is present in curd. It turns milk into curd. That is very important. Now, based on the mode of nutrition, bacteria can be further classified into two subgroups. One is the autotrophic bacteria and another is the heterotrophic bacteria. Autotrophic bacteria can be classified into further two subgroups, the chemoautotrophs and the photoautotrophs. Now, these chemoautotrophs, they are mainly marine and they break down complex chemical substances present in the ocean water into simpler substances and they derive their energy from that process only. Chemoautotrophs. Well, the photoautotrophs, they have chlorophyll, they can produce their own food by photosynthesis. Now, the heterotrophic bacteria can be classified into further three subgroups. The saprophytic bacteria, bacteria that break down dead bodies, bacteria that help in the process of decomposition of animal matter, plant matter, fungal matter, anything that is dead and organic. They break down into simpler molecules and they restore the nutrients back into the nature. This is done by the saprophytic bacteria. The symbiotic bacteria, rhizobium, where they are present in a symbiotic relationship with some algae, or in this case with some protists, even in with some fungi also. For example, rhizobium, or with the help of plants even. The rhizobium are also nitrogen fixing bacteria. They are present in the leguminous plant, the root nodules of the leguminous plants. So it is a symbiotic relationship with the plants. Rhizobium have. Parasitic bacteria are the disease causing bacteria, bacteria that cause various kinds of lethal diseases like cholera, like citrus canker, diseases like that. They are known as parasitic bacteria. For example, Vibrio cholerae, which causes the disease of Asiatic cholera. So this is all about kingdom Monera.